Can you hear and see me?
Yep. Hello, how are you?
Oh, I'm good. I'm excited that you're going to be giving us a talk today um, for the Global Virus Network, Forefronts in Virology.
Yep,
Now, you're a researcher at Menzies Health Institute, which is part of the Griffith University Gold Coast campus in Queensland.
yep,
Um, and you've been focusing on investigating the role of host factors, particularly MXRA8 and
yes,
FHL1.
yep, that's correct.
And near and dear to my heart in alphavirus diseases.
Yep,
So I'm looking forward to hear a lot more about that. And then you've also been working on a significant project in live attenuated vaccines for SARS-CoV-2. Um,
yes,
and hopefully we'll get to hear some of that as well. And then also been contributing to understanding and addressing a lot of public health challenges um, and have exhibited an intense dedication to the field of virology. So we're grateful to have you with us today. And without further ado, I'll let you share your slides and get started.
right, thank you, Maggie. Yep, can everyone see my slides? Yes,
Yes,
right, perfect. I can begin now, right?
yes, please.
All right, thank you, Maggie, for that kind introduction. So, I'm Wern Hann from Griffith University, based in Menzies Health Institute, Queensland. And the title of my talk today is FHL1 promotes chikungunya and onyong virus infection and pathogenesis implications for alphavirus vaccine design. So this is my presentation outline today. I'll be giving a short introduction about alphaviruses and FHL1. Next, I'll be showing some results on how FHL1 expression is upregulated by chickpea infection, followed by its clinical profile in knockout mice compared with wild-type mice. And I'll be also showing some histological analysis, the differences in cytokine, chemokine, and leukocyte profiles, the role of FHL1 in other alphavirus diseases, particularly Ross river, onyong, and myroviruses, and then also targeting FHL1 as prophylactic strategy. Then I'll, of course, will be just concluding my presentation. So alphavirus belongs to the family of Togaviridae and is divided into two different species, which is the atritogenic alphaviruses and the encephalitic alphaviruses. And as their name suggests, the encephalitic alphaviruses causes encephalitis symptoms, and the atritogenic alphaviruses causes musculoskeletal symptoms. I'll mainly be focusing on the atritogenic alphaviruses as that is the focus of my studies. And
Warren,
the
I'm
common,
gonna,
sorry.
sorry to interrupt. I'm noticing that you're in presenter mode instead of the
Oh, right.
presentation.
1 second. How do I change this?
I'm pretty sure, yeah.
Mm.
There you go.
Oh, well, okay,
Oh,
that
wait.
works.
Yeah, this works. This is better.
Yep, okay. As I was mentioning, my focus is on atritogenic alphaviruses, and some examples include the Ross River virus, chikungunya virus, myrovirus, and onyong virus. And out of these four, my focus today would be on chikungunya virus. So the chikungunya virus is mainly transmitted by the Aedes species, which is the Aedes aegypti and the Aedes albopictus specifically. It's associated with crippling persistent arthralgia, myalgia, arthritis, and myositis. There's a large outbreak observed globally and some The outbreaks include over 200,000 cases in the La Reunion Island in 2006. And in India, between 2005 to 2006, there was over 1.3 million cases and over 2 million cases in America from 2013 to 2016, around 15,000 cases in Thailand between 2018 to 2019, and more recently in Paraguay in 2022 and 2023, there's around 80,000 cases reported. And as of 31st March 2024, in Brazil alone, there's over 160,000 chickpea cases reported. Chickpea has been identified as a virus to cause the next pandemic, and the global impact of chickpea infection is estimated at over 106,000 disability-adjusted life years annually, and making it one of the WHO's top priority emerging infectious diseases, and is ranked 4th in public health need according to CEPI. Usually, chickpea is self-limiting. However, about 40 to 60% cases of infected patients develop into chronic phases, which limits day-to-day activities, and it reduces the quality of life in affected individuals. And it also imposes a heavy economic burden. The collective global cost of chikungunya between 2011 to 2020 is estimated at $46.7 billion USD. As I was mentioning, my focus is studying host factors that's associated with alphavirus pathology. And just to describe it briefly, the function of host factors in alphavirus pathology. So when an alphavirus, a mosquito, it bites the host, the virus will enter through attachment factors such as the MXRA8, the DC sign, and TIM1. And then as it enters the virus, it interacts with replication factors such as the FHL1. The virus will replicate, and then it gets into the virus gets in the host cells, and then it replicates. And then upon replications, all the cytokine responses and immune cell responses take place. And then all these will lead to in the case of atritogenic alphaviruses, your musculoskeletal symptoms, and for the encephalitic alphaviruses, your encephalitic virus symptoms. I'll be focusing on the FHL1. So FHL1 is a protein coding gene that belongs to the limb-only protein family, characterized by 4.5 highly conserved limb domains. And it's a causative gene of FHL1-related myopathies, which includes the reducing body myopathy, X-linked myopathy with postural muscle atrophy syndrome, rigid spine syndrome, and together with emeridriferous muscle disorder, this conditions affects the skeletal and cardiac muscle, causing joint deformities, and it restricts the movement of certain joints, muscle weakness, and wasting that worsen over time, and also heart problems. All this, if left untreated, will lead to increased risk of sudden death. So FHL1 was recently found to be involved in the translation and replication of chickpea and onion. In that paper, the authors extracted primary fibroblast and myoblast cells from EDMD patients, and these patients has mutated FHL1 gene, and then they infected them with chickpea, and then these cells, they show resistant to chickpea infections. And in that study, they found out that FHL1 actually interacts with the NSP3 region of chickpea. In the right, it's an oversimplified demonstration of what was done in the studies. So basically, chickpea NSP3 interacts with host FHL1, and then it multiplies and everything, and then it causes an acute chickpea disease. So for my study, the first part of my study, I had collaborators in Brazil. So we had some serum from chickpea infected patients and healthy patients. We collected them and then we measured for the expression of FHL1. And we also tried to replicate this in the murine model, in which we collected the serum and quadriceps from chickpea infected mice, and then we compare it to mock infected mice and also measured for the expression of FHL1. So what we found was the FHL1 expression increased in chickpea infected human and mouse. In the serum of chickpea versus healthy patient, we can see that in the healthy patient, there's a lower expression of FHL1, whereas in the acute and chronic chickpea patients, the level of FHL1 actually increased during infection. And this is again replicated in the mouse model, where we collected the serum at day 1, day 3, day 5. We compared the expression level and we found that the infected mice has higher level of FHL1. This is also reflected in the quads, and the image on the right depicts the confocal image of the muscles, where we showed that in infected mice, there's a higher level of FHL1 compared to the uninfected mice. Next, we move on to the next part, where we had generated a FHL1 knockout mice, and we compared the disease profile with the wild-type mice. So the wild-type mice and the knockout mice, they were infected with chickpea subcutaneously in the foot pad. And then we compared the disease score between these mice, where we look at the weight, the pain levels, and also the joint swelling of these mice. Next, we also collected tissues and measured them for virus titers, chemokine, cytokine responses, and then we look at some histopathology and leukocyte profiles. So what we saw was that in wild-type mice, the disease is much more pronounced compared to the knockout mice, as you can see in the green line here. At day 3 and day 7, which is the 2 peaks of a chickpea mouse model, we can see that the joint measurement, the joint swelling, it's much more pronounced in the wild-type mice compared to the knockout mice when infected with chickpea. And this is consistent also at day 7, where we can see the increased peak compared to in the wild-type mice compared to the knockout mice. Next, we look at the virus titer in the serum and day 1, 3, and 5. We can see that in the wild-type mice, the virus titer is consistently higher compared to the knockout mice. And then, of course, we also look at the virus titer in the quads at day 3 and day 7, and also the ankle at day 3 and day 7. And consistently, the virus titer is higher in the wild-type mice compared to the knockout mice. And this tells us that FHL1 is important for chickpea replication. Next, we look at some histological analysis. So we collected the quads at mock infected mice, and at day 7, which is the peak of chickpea disease in the mouse model, what we saw was that in the control, there was no differences between the infiltrated cell density and also muscle damage in both the quads and the ankles. Whereas at day 7, we saw that there's an increase in the infiltrated cell density in both quads and the ankles. And also from the panel, we can see that there's increased muscle damage in the wild-type mice. Next, we look at the cytokine and chemokine profile compared between the infected chickpea wild-type mice and also the knockout mice. And what we saw was in the profile that is highly related to the alphavirus pathology, we saw that it's consistently higher in the wild type mice compared to the knockout mice. Again, like it just tells us that FHL1 is important factor for chickpea replication. This is also reflected in the leukocyte profile, where in the T cells such as the CD3, CD4, CD8, macrophages, monocyte, and neutrophils, and also the NK cells, consistently it's higher in the wild type mice versus the knockout mice at day 7, in the quads at day 7 time point. But at day 3, although there's non-significance, we actually saw that the trend was slightly increased in the wild type mice. What we think that is that this is an early time point, and it's not like in the knockout mice there's no disease. It's just that the disease level is reduced. However, some replications is still taking place. So this could explain why that the knockout mice and the wild type mice, the differences in the leukocyte profile between these two mice is not that pronounced yet at day 3. However, by day 7, we can see that it is significantly increased in the wild type mice. Next, we also look at the differences in the leukocyte profiles using mass cytometry. So what is mass cytometry? So to put it simply, mass cytometry replaces the fluorescent reporters used in flow cytometry with metal isotopes, which are detected using cytometry by time of flight. And this can expand the number of parameters that can be detected simultaneously on a single cell to approximately about 40 to 60 due to the absence of signal overlaps between the metal isotopes. And what we saw was that in the lymph nodes and the spleen compared between the wild type mice and the knockout mice, the clusters of major cell types were identified on this UMAP plots based on heat maps of the expression level of fundamental cell markers, such as the CD3, CD4, CD8, CD11B, and CD19. And there's position shift on this UMAP plot. It represents the phenotype changes of the cells. So cell markers that principally contribute to chickpea-induced shifts includes the CCR7, the Ly6C in various cell types. And in the lymph nodes, this surface antigen were found to be increased on the T cells and B cell lymphocytes, as well as the CD11B myeloid cells in both the chickpea-infected FHL1 knockout and wild-type mice. However, while cells in the infected wild-type mice tend to maintain relatively high level of CCR7 and Ly6C expression over time, the levels were significantly lower in the infected FHL1 knockout mice, with these differences greater at day 7 compared to day 3. And the differential expression between the chickpea-infected FHL1 knockout mice and the wild-type mice was also observed in the spleen at day 7, although interestingly, in contrast to the lymph node, the Ly6C expression was significantly higher in the B cells and the CD11B myeloid cells at day 3 post-infection. And the profile of these surface antigen is in line with the lower disease symptoms observed in the chickpea-infected FHL1 knockout mice. Furthermore, the higher levels of Ly6C in the infected FHL1 knockout mice suggest that there's a potential novel role for the Ly6C and monocytes in the early stages of chickpea disease pathogenesis. Moving on, so we decided to look at the role of FHL1 in other alphaviruses, such as in particularly the Ross River, Myaro, and Onyong. So we had the wild-type mice and the knockout mice. We infected these mice with said alphaviruses. We look at the disease score, and then we, of course, measured the virus titer. So this is the role of FHL1 in RRV disease. Again, we took serum from patients, human samples. We compared the FHL1 expression between the healthy and the Ross River virus patient. And we saw that in Ross River patient, the level of FHL1 is actually increased. Next, we compared the disease profile between the wild-type mice and the knockout mice. And interestingly, we did not see any differences in the disease score. And this is also reflected in the virus titer obtained from the serum and the quads and ankles. Whereas in Onyong and Myro model, we saw that there was significant differences between the disease profile in Onyong. However, in Myro virus, we do not see any differences similar to Ross River virus. And these are reflected in the virus titer obtained from the serum and quads, and also the ankles, where in the Onyong profile, the virus titer in the wild-type mice is higher compared to the knockout mice, and there are no differences in the virus titer between the wild-type mice and the knockout mice in the serum, quads, and ankles at all time points in the Myro model. So what we did next was we fused the hypervariable domain of the NSP3 of Ross River, Myro, chickpea, and Onyong to eGFP, and we expressed this fusion protein in U2OS cells, and we perform a pull-down assay experiment. So what we saw was that the NSP3 of all four different viruses interacts with the G3BP1, which is an essential host cofactor for replication of atritogenic alphaviruses, and only the NSP3 of the chickpea and onion were able to interact with the FHL1 as shown in the pull-down assay. So this indicates that FHL1 is essential for chickpea and onion infection and disease development, but not for RRV or myrovirus. Next, we came up with the idea of targeting FHL1 as prophylactic measures against chickpea. So we designed a live attenuated virus where it does not react with the FHL1 region of the host. We infected these mice, we immunized these mice with the virus, and then we challenge it with various alphavirus. We measured the disease score, looked at the virus titer, and also did the PRNT assay. First, we looked at whether the live attenuated virus candidate design will cause disease in mouse. So we did the chickpea immunization, and then we compared its disease profile between the chickpea wild type and also another chickpea live attenuated chickpea vaccine, which is already licensed in human, which is the chickpea 3 delta 5. What we saw was that in our chickpea live attenuated candidate vaccine has a similar disease profile compared to the licensed vaccine, whereas the chickpea wild type mice the chickpea wild type virus compared to our vaccine candidate causes a significant level higher of disease in mouse. And this is reflected in the quads at day 3 and day 7 post-infection, where we saw that there's higher level of virus titer in mouse when infected with chickpea wild-type virus. But we do not see virus at all at day 7 post-infection. So this indicates that our live attenuated vaccine, chickpea FHL1 knockout, is safe for use. Next, we challenge these mice, immunized mice with various alphaviruses. In the chickpea model, we see that the vaccine actually protects the mice from chickpea challenge, and this is also reflected in the virus titer. Similarly, for onion, the mouse are protected from onion challenge when immunized with our live attenuated vaccine, and again, it's reflected in the serum when we measured the virus titer. And interestingly, despite not being essential, FHL1, despite not being an essential host factor for alphavirus replication of MXRA8 and Ross River virus, this chickpea FHL1 knockout vaccine candidate actually protects the mice against MXRA8 and Ross River virus challenge, where we see that in the disease model of mice model of MXRA8 challenge, there's a significant differences between the joint measurement when challenged with MXRA8 in the mock immunized mice compared to the immunized mice. And this is also reflected in the virus titer in the serum at day 1 and day 3 post-infection. For Ross River virus, we are not able to measure the disease score as it's age dependent. So post-immunization of this mice is already way past the age where we can accurately look at the disease profile of these mice, because they simply don't show disease when they are past a certain age when you challenge the mice with Ross River virus. But what we can see was at day 1 and day 3 post-infection, the mock immunized mice, the virus titers of the mock immunized mice at day 1 and day 3, when challenged with Ross River virus, is higher than the immunized mice when challenged with Ross River virus. So next, we also did a PRNT assay where we showed that the percent neutralization of the live attenuated vaccine. So what we saw was that the PRNT value of chickpea, RRV, MXRA8, onion, there's a high dilution factor where it suggests that this live attenuated vaccine can protect against chickpea, Ross River virus, MXRA8, and onion challenge. So in conclusion, the expression of FHL1 is increased during chickpea infection in human and mice. Virus titer recovered from chickpea-infected FHL1 knockout mice were reduced compared to chickpea-infected wild-type mice. The total cellular infiltrates were reduced in chickpea-infected FHL1 knockout mice compared to wild-type mice. And immune cells is reduced in chickpea-infected FHL1 knockout mice compared to wild-type mice. FHL1 is required for optimal chickpea and onion infection in mice, but not Ross River or MXRA virus. And a mutant chickpea virus targeting FHL1 can act as a potential live attenuated vaccine and can also induce broad spectrum protection against other alphaviruses. I'd like to acknowledge my supervisors, colleagues, and also family and friends. Thank you.
Fantastic work. Thank you for sharing your talk. I will remind everybody that they're able to post questions to the chat, and I will ask them. I have many questions. I already told you I'm a big alphavirus fan. What do you anticipate, or what do you hypothesize the role is across alphaviruses early in pathogenesis that you've picked up on?
Sorry, come again.
Sorry, what do you anticipate the role of specifically FHL across these different alphaviruses and what made it an ideal target?
So what we saw that FHL1, it's important in chickpea and onion, but not Ross River and Mayaro. And this suggests that there's an evolutionary mechanism behind this host factors. So as I've mentioned before, FHL1, it's responsible for many FHL1 related myopathies. And all these myopathies actually causes muscle weaknesses, which is
Right.
also the main target for alphavirus. So alphavirus, a tritogenic alphavirus targets the muscle. And what we anticipate is that due to some host evolutionary mechanism over millions of years, this causes the alphavirus to target the FHL1, which causes the increased level of expression in FHL1 in cells to make up for the so like when a cell, you have FHL1, so when it's targeted, the cells sense that, okay, we need to make more FHL1 that causes the increase in FHL1 expression. Does this answer your question?
Yeah. I think the other thing that's interesting about this area of work is it reminds me of other inherited collagen diseases and the differential impact to disease severity that has. So it was intriguing to see that there might be some infection resistance in these rare different types of heritable conditions.
Yeah.
And so, yeah, so I guess, you know, these viruses are spreading further and further due to a number of factors. Are the incidence of FHL conditions localized to specific regions, or do you see a higher prevalence for these in regions in which the viruses are already commonly circulating?
Sorry, I don't quite follow your question there. So do you mean that the virus targets just high level of organs with high level of FHL1 expression? Is that what you are trying to?
No, I do see. So I was curious if the distribution of people with familial if that condition is prevalent in specific areas of the world, and if you would anticipate more people being susceptible or resistant to the arthralgic form induced by the condition, or if that's known.
No, I don't think so, because we know that people with FHL1 disorder have some resistance to alphavirus infection, as shown in the paper, the paper that discovered the link between FHL1 and tritogenic alphaviruses. So I don't see the link, actually. I don't.
No, that's reasonable. I see a question in the chat from our president, Sven Rahman. Thanks for your 4 a.m. contribution from Australia. Oh my goodness. And what therapeutic strategies are suggested by the knockout mice dynamics?
So for therapeutic strategy, we do see that if we can somehow find a way to target FHL. FHL1 is an intracellular host factor, so that there's some difficulties in delivering in targeting this host factors as a therapeutic measure. But if we can find a way to do it, and which we are trying to do some studies in it, deliver this antibodies to target FHL1, we can see that it is able to, we predict that it is able to, in a way, stop the disease from developing to a chronic phase. So that's how we see it. But of course, it's still subject to studies as, again, it's an intracellular protein. So that the design of this therapeutic can be quite challenging, and we are also mindful of any other side effect that comes along by targeting FHL1, as it is the causing gene for many FHL1 myopathies. Yes.
No, that's an excellent point.
Yes.
We have another question in the chat from David Martinez. Have you looked at the durability of the antibody response in the mice given the live attenuated chikungunya mutant virus used as a vaccine?
We have not looked at it long term, but what we know is that after vaccinating these mice with our live attenuated vaccine candidate, up to 3 months post-vaccination, we still are able to detect some titers in the mouse. But we did not publish this data, and yeah.
Do you have any, and this is a follow-up question from David, do you think this durability compares to natural infection in the mice infected with the wild-type chikungunya strain?
We believe it does, actually. We are confident it will, due to the high level of PRNT titers that we actually obtain. Like the level goes up to, the dilution goes up to thousands, which is actually relatively high, like PRNT 75. Yep. So we believe it's comparable to natural infection.
That's fantastic. I'm seeing a question from Jonathan Eubank. Are any other LIM proteins targeted by alphaviruses?
Sorry?
Are any other LIM proteins targeted by alphaviruses within the same family?
Not to our knowledge, no. So FHL1 is the only LIN family protein that is actually related to alphavirus.
Oh, that's interesting. And I wonder if you had noticed some really interesting dynamics in the immune repertoire and the response, but those percentages, while significant, do you have any sense of if they're clinically significant or what those impacts would be? I guess I was thinking about the differences you saw in like NK cells.
Right. So with alphavirus, the main culprit is actually the macrophages, but of course NK cell plays some role. So yes, actually, like when host is infected with alphavirus, all these immune cells go into overdrive, and this is what causes the disease symptoms in the muscles. And I'm not quite understanding that question, actually. So does it relate to FHL1 knockout as well? Like
Right. Are you seeing different discrepancies that are associated specifically with that?
Yes, actually. So what we saw was in the knockout mice, this levels of these immune cells is actually lower. So to a certain level, it does, due to the lower expression of this macrophages, NK cells, monocytes, and in the FHL1 knockout mice, this could have led to reduction in the diseases compared to the wild-type mice. But at the same time, we can also argue that because in the knockout mice, there's no FHL1, so the chickpea is not able to replicate efficiently. So that also leads to reduced virus, and lower virus leads to reduced disease. And because of that, there's lower immune responses. So to put it bluntly, we don't know. Yeah.
No, that makes complete sense. I don't see any other questions in the chat. I really appreciate you giving us this fantastic talk, and I want to thank you for educating us on this topic. And without further ado, we'll let you go and hope to see everybody next time.
Yep. Thank you for having me. It's been a pleasure.